to another screencast. This is on the Krebs cycle and this little intermediate step in there as well. So the goal is here to describe and explain the reactants, products, location, and purpose of the Krebs cycle. Again, reactants, products, location, purpose of the Krebs cycle, the two subphases of the Krebs cycle, and then the purpose, again, of NADH, which we keep hitting. We hit with glycolysis as well. Explaining substrate level phosphorylation maybe, and then we'll get into this oxidative decarboxylation, which is a mouthful to say the least. But this is descriptive of what's happening in the Krebs cycle. So just to put a big picture here, where are we? We've already addressed the glycolysis here, so now we're going to take the pyruvate from glycolysis here and enter this Krebs cycle. So we're talking about all of this stuff right here. This is where we are with this screencast. Now, in class today we had this one, pardon me, that's a little bit more detailed, and you got this box that's here. So you see that this would be pyruvate, and then here's the Krebs cycle itself. But there's this little intermediate step that your textbook addresses that's sort of important. So we're going to start right there first before we go into the Krebs cycle proper. So this is that intermediate step. What's happening is that pyruvate from glycolysis is a 3-carbon compound. And this is all out here in the cytosol. So there's a transport protein that's going to move or transport this pyruvate across the outer membrane, through the intermembrane space, across the inner membrane, into the matrix of the mitochondrion. So this is the matrix of the mitochondrion. And when it enters the matrix of the mitochondrion, there are enzymes in here that are going to do a few things to the pyruvate, right? Thing 1, thing 2, and then thing 3. So this is the intermediate step. Pyruvate is going to first be decarboxylated. And again, from a previous screencast on fermentation, we know that the decarboxylation part is removing a carboxyl group. So here's the carboxyl group. And so we're getting, in a decarboxylation, we're getting CO2, 1 carbon, 2 oxygens. Well, here's 1 carbon, there's 2 oxygens. So that's not quite CO2, but what we're going to do is decarboxylate, but then we also have to oxidize this. So if we oxidize this, that takes that hydrogen away. And so we can do an oxidative decarboxylation. But when we oxidize the pyruvate, right, we notice that right here, this is showing it's sort of already oxidized, right? But we could assume that there's a hydrogen right there. So we're going to take this CO2 group off, and we're also going to oxidize it. Well, if we oxidize something, something has to be reduced. So what are we going to do? We're going to reduce NAD+, right? So this is the reduction here, right? And the oxidation is this way. So we're going to oxidize and decarboxylate. Oxidize and decarboxylate the pyruvate. And when we do that, we generate CO2. We generate NADH, we generate NADH, right, which is a good thing. And then we're going to generate this 2-carbon compound over here. Well, wait a second, 2 carbons, 3 carbons. Where did the carbon go? Ah, that's right, it went right here. Here's the 1 carbon that left. The third thing we do is we stick this thing called coenzyme A on there, and we get this intermediate molecule called acetyl coenzyme A. What is coenzyme A? It's coenzyme A. Don't worry about it. We can talk about it later if you have more questions about it. But this coenzyme A thing here gets stuck on this. So the point here is that this acetyl coenzyme A is a 2-carbon compound. And this is going to the Krebs cycle proper. Right? So pyruvate over here does not enter the Krebs cycle proper. It's this intermediate acetyl coenzyme A thing that enters there. So we oxidize and decarboxylate. Incidentally, this carbon dioxide that's here is going to diffuse out of the cell. Diffuse out of the cell, right? And if you are a mammal like us, then it's going to diffuse out of the cell, it's going to diffuse into the bloodstream, it's going to then diffuse from the bloodstream into your lungs, and you're going to exhale it. If you're a plant cell, and you're a root cell or a stem cell or a leaf cell, this CO2 is just going to diffuse out of the cell. If it's in the leaf, it's going to diffuse out of the stomata. Right? So this is the source of all the carbon dioxide that is a byproduct of respiration. So CO2 there we have now completely oxidized any organic molecules that came in. Remember, going all the way back, that we had we started this whole thing with glucose, which is an organic molecule, and we're going to oxidize glucose. Right? The complete oxidation of glucose gets you CO2. When you oxidize organics completely, these organics, CHO, right? The complete oxidation of organic molecules leaves you with CO, right? And you do some stoichiometry and you end up getting CO2. So this intermediate step, there's that. Little graphic organizer to sort of summarize that. What are all the boxes? Right? Let's go talk about the Krebs cycle proper now. So we now know that this intermediate step is producing CO2 and acetyl coenzyme A, and it's producing some NADH. Here's the Krebs cycle proper. Right? So let's talk about that. Look, here's our 2-carbon acetyl coenzyme A going into this Krebs cycle. Right? So this is what we're going to address here in the next couple of slides. This is the nitty-gritty detail. And again, you do not need to know all these intermediates by any stretch of the imagination. When you grow up and become a biochemistry major in college, then you can do it. Right? So here's a 2-carbon compound. This is a 4-carbon compound. This then is a 6-carbon compound. The only reason I'm adding any details right now is just to show you some simple math. Right? 2 plus 4 equals 6. I'll do it again. 2 plus 4 equals 6. This first molecule right here, this citrate, is also sometimes known as citric acid if it was not ionized. And this is the other name for this cycle, is sometimes referred to as the citric acid cycle. And why? Because the first molecule that's made in the cycle is the citrate molecule. Regardless, the Krebs cycle is going to make a bunch of organic intermediates that are going to be over here. So this is an organic intermediate. An organic intermediate molecule right here, this isocitrate. What's going to happen to this? Well, look, here's isocitrate. This is a 6-carbon molecule. Here's alpha-ketoglutarate down here, which is a 5-carbon molecule. Okay, 6, 5, what's the difference? 1. Well, where did the carbon go? Wait, we saw this before in the previous slide, right? We had a 3-carbon pyruvate. We got a 2-carbon acetyl coenzyme A. Where did the carbon go? It was lost as carbon dioxide. Oh, that's going to happen again right here. Right? I'm going to decarboxylate, and I'm going to oxidize and reduce. Right? So this is an oxidative decarboxylation. Then look, we do it again. 5-carbon gets me this 4-carbon thing down here. I'm going to oxidize and decarboxylate. Oxidize, decarboxylate. I'm generating CO2. I'm generating NADH. I'm generating CO2. I'm generating NADH. What am I doing? I'm oxidizing organics, reducing NAD+. I'm decarboxylating. I'm decarboxylating. That gets me this 4-carbon molecule down here. Let's continue all the way back around. Okay, 4-carbon. This is a 4-carbon, and this is a 4-carbon, and I get this oxaloacetate thing back up here. That's another 4-carbon. What we also see along the way is that we're going to, over here, when I go from succinate to fumarate here, I'm going to do a redox reaction right there, where I'm going to oxidize succinate to fumarate, and I'm going to reduce FAD to FADH2. Then I'm going to, up here, here's another redox reaction up here. I'm going to oxidize malate to oxaloacetate, and I'm going to reduce NAD+ to NADH. So I'm getting a redox and a decarboxylation. I'm getting redox and decarboxylation. I'm getting redox. I'm getting redox. And then at the very bottom of this, I'm also, in this step down here, going to be generating ATP. So as this Krebs cycle spins round and round, we're doing several different things. Let's go look at the title now. This may make sense. The Krebs cycle oxidizes and decarboxylates organic intermediates. NAD+ and FAD are reduced ADP is phosphorylated, and CO2 is released. Every acetyl coenzyme A that comes in here, this spins around one time. So here's a much more simplified version of that. Starting at the top again, here's the intermediate step that we're showing you. Right? So we start with 2 pyruvate molecules that are 3 carbon. Right? So all this is happening twice, right? It's happening twice for 1 molecule of glucose that started all the way back in glycolysis. So 1 molecule of glucose gets us 2 pyruvates. Those pyruvates are oxidized and decarboxylated. I don't know why this isn't writing. Oxidized and decarboxylated to get coenzyme A. This 2 coenzyme A enters the Krebs cycle. Notice you don't have to know any of the intermediates here, but what you do need to know is that there is another decarboxylation here. There is a redox happening here. There's a redox happening over here. And there's a phosphorylation happening here to generate ATP. So for every 1 molecule of glucose that went in, I'm generating carbon dioxide. But really what I'm generating is NADH, which is important, and I'm generating ATP, which can be used, and I'm generating FADH2, which is important. So this ATP down here is used by the cell. This FADH2, where's it going? It's going to go to electron transport, oxidative phosphorylation. These NADHs, where do they go to? They're going to go to electron transport chain oxidative phosphorylation. Where's all this happening? This is all happening in the matrix of the mitochondrion. In matrix of mitochondrion. Matrix of mitochondrion. Okay. So again, we're dealing with here this part of the diagram. We're getting this happening right here. Intermediate step, Krebs cycle proper. CO2, CO2, ATP, NADH, FADH2, NADH. These things are going to go to electron transport chain. This CO2 lost, diffused out. There is another graphic organizer. The intermediates here you don't need to know, but again, this is going to be a 6 carbon. That's a 6 carbon. So just do this around 5 carbon, and then that's a 4 carbon, and then these guys are 4 carbons all the way back around because that's a 6. That's a 4 carbon there. So what goes up here? This is not showing you the intermediate step. This is Krebs cycle proper. Okay. We'll do this in class here to kind of add up and do the itemizing. But they're going to put numbers in here. Right? So if you want to try to pre-fill that, that should be that'll give you a good idea if you really understand what's going on. So it might be a good idea to sort of do that as a practice. I just came across this guy's cycling jersey. It's pretty funny down here that the name of the cycle company, the name of the bike shop that sponsors them is Krebs. And then down here, if you zoom in, they have this little Krebs Cycle. It's pretty funny. So they're producing CO2 and then miles per hour, right? And so just ATP energizing. This is what like nerds, creative nerds do with their time, I guess. So anyway, there's some blanks for you to re-summarize, and hope that's helpful. Bring your questions to class.